Good morning, welcome Shem Stars. In previous video we studied DNA fingerprinting and important terms related with DNA fingerprinting. So in this video we are going to learn about technique of DNA fingerprinting and applications of DNA fingerprinting. As we know, DNA fingerprinting is a quick way to compare the DNA sequences of any two individuals, and this technique was initially developed by Alec Jeffrey. He used a satellite DNA as a probe that shows very high degree of polymorphism, and it was called as variable number of tandem repeats, that is VNTR. Since DNA from every tissue such as blood, hair follicle, skin, bone, saliva, sperm, etc. from an individual show the same degree of polymorphism, they become very useful identification tool in forensic applications. There are 8 steps for DNA fingerprinting. Step 1. Isolation of DNA. DNA must be recovered from cells or tissues. Only a small amount of blood, hair or skin is needed to isolate DNA. Step 2. Digestion of DNA by restriction endonucleases. Enzyme restriction endonucleases. The DNA is cut into fragments using restriction enzymes. Each restriction enzyme cuts DNA at a specific base sequence. Step 3. Separation of DNA fragments by electrophoresis. Fragments are separated on the basis of size using a process called gel electrophoresis. DNA fragments are injected into wells and an electric current is applied along the gel. Step 4. Transfer of DNA on nylon or nitrocellulose membrane. The DNA fragments are transferred to a nylon sheet by placing the sheet on the gel and soaking them overnight by the process southern blot. Fifth step of this technique is probing or probe labeling. Adding radioactive or colored probes to the nylon sheet, which is complementary to target sequences. Each probe only sticks to one or two specific places on the sheet. Step 6. Hybridization. Labeled probe DNA should be hybridized with the complementary sequences located on nylon membrane for the detection of position of later. Membrane is washed to remove non-specific binding and clearing of the background. Next is step 7. Autoradiography. To detect the sequences in genome bound with the hybridized radioactive probe on membrane. Technique involves alignment of hybridized membrane with X-ray film. The X-ray film alter its development shows multiple number of bands that looks like barcodes and known as DNA fingerprints. And the last step of this technique is interpretation of band patterns. Analysis of band patterns of different individuals, comparison of position of bands. Computer software are also available for the analysis of DNA fingerprints. So here are the results. We can compare band patterns of two individuals which are shown with the two different colors. Now let's talk about applications of DNA fingerprinting technique. Human DNA analysis. It is used to test for genetic disorders. DNA fingerprinting used to identify individuals. Human Genome Project, which we have already discussed in previous presentations. It is used for mapping of human genome. Gene therapy. An absent or faulty gene is replaced by a normal working gene. So children, this is all about DNA fingerprinting technique. I hope you must have understood, and this is the last topic of chapter 6, molecular basis of inheritance. Thank you, Shem Stars. That's all for today. You can drop your queries on Google Classroom. Take care.